Hello everyone. Today we will study about commonly used laboratory animals in experimental pharmacology. Let's start with the purpose for the use of experimental animals. So the experimental animals are very important in case of pharmacological study, and they are generally used for investigating safety and effectiveness of drugs, cosmetics and other households made for human use. So you can see that apart from the drugs, they are also used for investigating the safety and effectiveness in cosmetics and other households which are used for human use. They are also used for investigating the effectiveness of drugs before testing on humans. Since the animals which are used on experimental pharmacology, they are also called as preclinical studies. Since the life of a man or human being, it is very precious, we cannot directly study any chemical compound on human being. Therefore the drug, it is tested on animals first to confirm their safety. Then only we can test the drug on human being. Let's see about the selection criteria for experimental animal, that on what basis you are selecting different type of animals. The animals which are going to be select should have physiological similarity to human being. For example, if you want to conduct a study on heart or lungs, it should have similarity with the human. As you can see in the picture, the brain, heart, lungs, kidney, intestine, all the organs are similar to that of the rat. So whatever study which we want to perform on brain, lungs, heart, liver, intestine and kidney, we can perform on rats first. And uh, uh, if we will found a safety and efficacy on the rat, we can perform the same study on human beings. In addition to the physiological similarity, it should also be easy to evaluate a process under investigation. For example, if we are performing an anti-diabetic study on an experimental animal, we should choose such animals that it will be easier for the investigator to withdraw a blood. For the anti-diabetic study, if we will choose a mice, which is a small animal, like it is around 25 to 30 gram in weight, whereas the rat, the weight of the rat, it is between 250 to 300 gram. 300 grams, so it will be easier for the investigator to withdraw a blood for the blood sugar, to check the blood sugar level in anti-diabetic studies. So it should be also easy to evaluate a process under investigation for the investigator while selecting an experimental animal. Now let's see what are the environmental conditions which should be maintained. The experimental animal room is adjustable to meet the special requirement of the investigation and the need of the animal species. The experimental animals are quite sensitive. A small change in temperature or humidity may lead to change in physiology, which may finally hamper or alter the results of the experiment. So generally a constant and comfortable environment is required to ensure the health of the experimental animal and to get the meaningful, reproducible results. For example, one day the AC is working properly and you have got the nice results. Another day the AC is not working, the environment has changed, then you may not get the same results which you have got earlier. So maintaining the constant environment, it is very important. Let's see which are the important parameters required to maintain particular environment. They are temperature, humidity, ventilation and light. Let's see each parameter in detail. First, it is temperature. A sudden change in temperature may harm laboratory animals. Generally, for small laboratory animals, the range of temperature should be between 20 to 30 degrees Celsius. If a small change in temperature occur, for example, for mice, the temperature has rise, the it may lead to the death of the animal. So it is very necessary to maintain a constant temperature, and to maintain this constant temperature, air conditioning system should be installed in the animal house facility. Next parameter, it is humidity. Fluctuations in relative humidity can precipitate illness such as respiratory illness. During the raining season, during the rainy season, relative humidity can be increased. For that reason, we may use dehumidifiers to control the environment. A normal humidity which will be required for the experimental animal is around 50%, but they can also tolerate the in the between the range of 30 to 70%. Next environmental parameter, it is a ventilation. Animal facility should be ventilated properly. We can install exhaust fans to maintain proper ventilation. Preferable to use a total air exchange system. For example, HEPA filter can be used for the air exchange system. Air conditioning system can be used to provide a stable environment. Next parameter, it is light. An animal facility should provide good and uniform glare free light. Glare free light means it should not glare on the eyes of the animals, and neither it should be too dim. And experimental animals require a 12-hour dark and 12-hour light system. For this, we it is necessary to install an artificial light into the animal house facility, and this 12-hour light and 12-hour dark cycle, it can be maintained with the help of timers. With the help of timers, after the 12-hour light has been completed, the timer will switch off the light automatically. Like that, a 12-hour light and 12-hour dark cycle can be maintained. In addition to environmental conditions, we also require to provide proper food, water, bedding material, and cage to the experimental animals. Prepared pallets can be provided to the rodents like mice, rats, and hamsters. But in case of rabbits, vegetables should be provided. Water ad libitum is mostly used. Bedding material, corn cob, and husk are mostly used. Cage are made up of aluminum box with good capacity. Lids are made up of steel. Now let's see the classification of experimental animals. They are widely divided into three types: rodents, non-rodents, and miscellaneous. In rodents, a small sized animals are classified. For example, rats, mouse, guinea pig, and hamster. Non-rodents contains rabbits, dogs, cats, monkeys, and pigs. In miscellaneous, frogs, zebrafish, pigeon, and chicken can be considered. Frog is banned nowadays, but zebrafish, it is widely used animal for the many type of experiments. Pigeon and chicken, the ileum can be taken for the in vitro studies. Yes, from this slide we will be studying each animal individually. So when we discuss about each animal, you should remember the biological name, unique food habits if they have any unique food habits, and for which experiment that animal is being used. So first animal, it is rat, which is also the biological name is Rattus norvegicus. The size, it is between 180 to 300 gram. They are sturdy and can be used for long experiments. For example, the experiments which required 6 months or more, the rat can be used. They have good sensitivity to many drugs, and they can be used for behavioral studies on CNS, anti-conversion studies, anxiolytic studies, CNS stimulants, CNS depressant activities, sleeping, sedative and hypnotic activities can also be done. And anti-diabetic studies can also be performed. Gastric acid secretions, hepatotoxicity study can also be done on rats. Bioassay of hormones like oxytocin, insulin can also be done on rats. Next experimental animal, it is mouse. The biological name is musculus. They are smaller in size, around 10 times smaller than rats. They are between 10 to 25 grams in weight. They have good sensitivity to small dose of drugs, similar reproductive and other system to human, and they have low maintenance costs. Low maintenance costs as they require less amount of food, less amount of water, less amount of drug to induce any type of disease, and they also require a small space for their living. In a one cage, around 10 to 15 mice can be placed, whereas in a single cage only 4 to 5 rats can be kept. The experiments performed on mice, it is similar to that of the rats, that is behavior studies on CNS, anti-diabetic studies, toxicity studies, it is unique, and genetic and cancer studies can also be performed on rats. What are these toxicity studies? Uh, Before administering any dose which is not known, we perform toxicity studies on smaller animals to know what should be the dose which can be administered to the human being. Next experimental animal, it is guinea pig. The biological name is Cavia porcellus. They are small in size, around 250 to 300 gram. They eat green vegetables and roots. They are highly sensitive to histamine. Since they are sensitive to histamine, it can be used in histamine bioassay, study of diphtheria vaccine, TB vaccine, activity of local anesthetic, hypersensitivity study, activity of bronchodilator can also be done on guinea pig. Next experimental animal, it is hamster, also called as Mesochrysatus auratus. They are small in size, around 60 to 100 gram in weight. They are commonly used species. They are in two species. They are in two types, the Chinese and golden species. The one which is in image is of a golden species. The experiment which can be performed on hamsters are study of viruses such as influenza, oncovirus, and vaccine study. Smoke inhalation studies can also be done. Study of mutagenic and carcinogenic agents can also be performed on hamster. Next experimental animal, it is a rabbit, also called as Oryctolo Oryctolagus cuniculus. They are larger in size, around 2 to 5 kilogram in weight. They also eat green vegetables and roots. The experiments, the large volume blood study can be performed on rabbits as they contain a larger amount of blood. Study of parenterals can be done. A pyrogen testing can be done on rabbits. Anti-diabetic activity which require a large amount of blood withdrawal can be performed on rabbits. Pyrogens are the microorganism which basically increases the body temperature. These type of studies can be performed on rabbits. So we have seen the rabbit. Likewise, we can use the dogs, monkeys, cats for the same activity at larger scales. The dogs, cats, and monkeys, they are not used at institute level or college level. They are mostly, the studies on them are mostly done on larger scale. Next animal is zebrafish. It is a novel model widely used. The size, it is between 3 to 6 centimeter. They also have less maintenance cost because they only require a fish tank and a small amount of food is required, and it is possible to perform high throughput study. High throughput studies are the one which require larger number of animals for the experiment. Since the cost it is less, we can perform high throughput study on zebrafish. The experiments which can be performed on zebrafish are embryogenic study, teratogenic activity of drugs, Alzheimer disease, anti-cancer activity, etc. So I have done with my today's session. I have also included some Viva and synopsis questions which can be asked during the exams. The next is the references. Thank you for watching.